Hello everyone, welcome to Plus One Biology. I hope all of you have opted biology with a great ambition to learn biology and become something under various fields of biology. Okay, you know me and this is our first class and we are beginning our Plus One learning chapter wise from the NCERT syllabus. Okay, and you know that our curriculum is revised and we have already discussed our revised curriculum in the last session, and now all of you are familiar and we have many chapters reduced and many topics reduced. And anyway, from the first chapter, the living world, we have very few topics to discuss. So I am beginning with the second chapter, biological classification. Okay, so the chapter is biological classification. In our interaction session and also in the bridge course, we have discussed that biological classification is a section which is coming under taxonomy. Okay, you have the basic ideas about the taxonomy, various types, branches of taxonomy, everything. Anyway, you know among the biological classification systems, you know the two kingdom classification introduced by Carolus Linnaeus. Okay, and he classified organisms into Plantae and Animalia. You know that, studied already, and its session is already over. Then still we can just have a revision. And you know the three kingdom classification. Okay, what is done here in the three kingdom classification is a third kingdom has introduced to separate what? All unicellular form. Okay, the new kingdom added was Protista, which included all unicellular organisms. That means from Plantae and Animalia, irrespective of whether it's plant or animal group, unicellular members were placed under kingdom Protista by Ernest Haeckel. You have studied it. And in the four kingdom classification, you have learned the next kingdom introduced was kingdom Monera. Okay, according to three kingdom classification, all unicellular forms were under Protista, kingdom Protista, whereas kingdom Monera separated unicellular prokaryotes. All prokaryotes were placed under this, among this unicellular forms. Okay, now Protista has only unicellular eukaryotes. Under four kingdom classification, this Protista has only unicellular eukaryotes. In order to place prokaryotes, there is separate kingdom Monera. And the most accepted five kingdom classification by Robert Whittaker, he has introduced the new kingdom which is fungi. Okay, from where it has come? The fungi, the kingdom fungi were earlier placed under, it was earlier under Plantae. Plantae include, Thallophyta include algae, okay, fungi like things. But kingdom fungi was separated by Robert Whittaker. Robert Whittaker separated kingdom fungi due to the cell wall nature and nutrition. That means the fungi which were initially under Plantae they were not similar to plants. They were not similar to plants in nutrition. They were not similar to plants in the cell wall chemistry of the cell wall. That means fungi had, instead of cellulosic cell wall, fungi have chitinous cell wall. And instead of phototrophic nutrition, fungi showed heterotrophic nutrition. Because of these two reasons, fungi were given a new status of kingdom status by Robert Whittaker. So Robert Whittaker introduced the fifth kingdom fungi, and he placed fungi and he has separated fungi from plantae because of the two major reasons. Because fungal cell wall is not, it was not similar to, it is like plants, it was not similar to plants, it was chitinous, made up of chitin. And also fungal cell wall, I mean fungal nutrition, and also fungal nutrition was heterotrophic. Okay, because of these two reasons, he has placed fungi separately into the next kingdom. Okay, and anyway, the 5 kingdom classification strategy we adopt now, and there are many characters considered in this classification, not only morphological feature. The 5 kingdom classification was based on morphology, anatomical feature, then cytological feature, mainly cytological feature. We can say the cell wall nature, okay, the chemistry of the cell wall. Then, and also it was taken into account the mode of nutrition. Okay, which type of nutrition was taken into account for the classification. Okay, more than this, all things, phylogeny also was considered as a criterion for classification by Robert Whittaker. So the 5 kingdom classification is more or less accepted even now because it has separated the complete organisms in the universe under 5 kingdom and considering almost all features like morphology, anatomy, embryology, cytology, then mode of nutrition, phylogenetic relations, like. Okay, so your textbook starts with, I mean the biological classification starts with various types of classification which we have already discussed in our previous class. So let us begin with Kingdom Monera. Today we do Kingdom Monera, the introduction session. Okay, let's move on to Kingdom Monera. Okay, and after learning from last day's class, okay, I mean the different classification strategies and today's class, what are the ideas you have about Kingdom Monera? What is meant by Kingdom Monera? Who has introduced this kingdom? You know, it's introduced by Copeland. Okay, and you know Kingdom Monera consists of what type of organisms? Unicellular organisms, which are prokaryotes or eukaryotes? Unicellular prokaryotes. So Kingdom Monera consists of unicellular prokaryotes. What do you mean by unicellular? That means the organism has only one cell. Okay, what do you mean by prokaryotes? They don't have a true nucleus. A true nucleus is absent. Instead, it has a genetic material which is seen scattered in the cytoplasm. There is no membrane bound nucleus present, and this is what is called nucleoid. Okay, something oid means something less than a nucleus. No true nucleus. Pro means primitive, carrion means nucleus. So these organisms, Monerans are prokaryotes, mean they have no true nucleus. Studied from the basic, 7th or 6th standard onwards you have studied this. So Kingdom Monera consists of organisms which are unicellular, only one cell. The organism means only one cell, and they are prokaryotes, means they don't have, the single cell does not have what? Nucleus. Instead, its genetic material remains scattered in a region called nucleoid. Okay, so that is about this. Organisms are called bacteria. So bacteria are the only members under Kingdom Monera. Is there any other group under Kingdom Monera? No, only bacteria are there under Kingdom Monera. And the details of a cell, its nutrition, everything we are going to learn. Before moving to its structure, just tell me where are the bacteria seen? Where we can see the bacteria? If you take a handful of soil, salt water, then well water, then air, Then, and if there is any type of diseases, it enters our body. And naturally there is E. coli in our body. That means the bacteria are said to be, or the Monerans are said to be cosmopolitan in distribution. These are cosmopolitan in distribution. Cosmopolitan in distribution means seen everywhere. Okay, see, the bacteria can even survive in extreme climates, extreme habitats. Okay, I mean the habitat extremes like hypersaline regions, ice cold region, permafrost regions, and our intestinal region inside other organisms. Like extreme climates also the bacteria can survive. The reasons, everything we discussed. Okay, then come to the first topic, morphology of bacteria. Structure, shape. Okay, you know the size of the bacteria. Okay, its size is in few micrometers size only. Okay, so the morphology of bacterium, when we discuss, we can see bacteria in four common shapes. Okay, some bacteria are spherical. Some bacteria are spherical. They are known as cocci. Singular term is single. If it's one, the term is coccus. Okay, whereas some bacteria are rod shaped. Okay, such bacteria are known as bacilli. Singular term, if it's one, it is bacillus. Okay, there are some bacteria which are comma shaped. Okay, this type of bacteria which are comma shaped are called vibrio. Comma shaped bacteria are called vibrio. If a singular, we call vibrium. The term, singular term is vibrium. Okay, and the next shape, spiral shaped bacterium. Okay, spiral shaped bacterium is known as spirillum. Plural term, spirilla. Okay, spiral shaped bacteria are spirilla. Singular term is spirillum. So you can see here four different shapes of bacteria. I think you need not need better clarity because from the shapes is clear. Then see, a spherical round shaped bacterium is called coccus. A rod shaped bacterium is called bacillus. A comma shaped bacterium is called vibrium, and a spiral shaped bacterium is called spirillum. Let us discuss one each example for each type. All you are familiar. Here example is streptococcus. You are familiar. You have studied pneumonia causing bacterium. Streptococcus is an example. Species name I am not giving now. Then bacilli. Example lactobacillus. You know the species name even. Still now you no need to learn. Lactobacillus is an example of rod shaped bacterium. Comma shaped bacterium. Example Vibrio cholerae, which causes cholera. Comma shaped bacterium. Example is Vibrio cholerae. Okay, Vibrio cholerae. Then example for spiral shaped bacterium is Rhodospirillum. It is Rhodospirillum. Okay, example for spiral shaped bacterium is Rhodospirillum. So these are the major shapes. Okay, morphologically bacteria, monerans are seen in these four major shapes. Simple fact, study one each example. Rhodospirillum, Vibrio cholerae, Lactobacillus and Streptococcus also. Okay. How are the bacteria able to move? Most of the bacteria possess what? Flagella. Okay. The arrangement of flagella also different. Some bacteria possess flagella all over the body surface. Some bacteria. Some contains only one flagellum at one end. The different name is there, amphitrichus, polytrichus, like so many names are there. Then it may be present on both sides. Single flagellum on both sides. And also a tuft of flagellum on both sides possible. Okay, this way different flagellar arrangements are seen in bacteria. Anyway, keep in mind, most of the bacteria possess flagella as locomotory appendages. Okay, and then come to the nutrition of bacterium. Nutrition of bacterium. First we have discussed the morphology and you know the size also. Okay, micrometer size and average size also you have studied. Then regarding the nutrition of bacterium, what type of nutrition is shown by bacterium? Bacteria show both autotrophic and heterotrophic nutrition. That means some bacteria are autotrophic whereas some bacteria are heterotrophic. There are bacteria which are autotrophic and also there are bacteria which are heterotrophic. Autotrophic bacteria can prepare their food by themselves. They are preparing their food. Okay, mainly two types are there. Photoautotrophic means which prepare food using sunlight. Photoautotrophs. Then the second type are chemoautotrophs which prepare food by deriving energy from chemical reaction, not from sunlight. Okay, so autotrophs prepare their food by themselves. Okay, they are nutritionally independent. Okay, they can prepare their food. And photoautotrophs prepare their food using sunlight, solar energy. Whereas chemoautotrophs prepare food using energy from chemical reactions taking place in the body, in the cells. Okay, then heterotrophs or heterotrophic nutrition. These bacteria are dependent on others. They cannot prepare their food by themselves. They are dependent. Okay, which mainly bacteria which are dependent on other living organisms show parasitic nutrition. Okay, they are dependent on living host. They derive nutrition from living host organism. And the next category, decomposers. Okay, decomposers are dependent on dead organism for food, not from the living host. And one more category is there, symbiotic. Some bacteria are symbiotic. Okay, which makes or which are involved in a mutualistic relation with a host. Anyway, regarding the nutrition of bacterium, autotrophic, heterotrophic. Autotrophic can be self-preparing using sunlight or chemical energy. Photoautotrophs, chemotrophs. Then heterotrophic nutrition, which are the bacteria which are deriving their nutrients from a living host, are parasites. Whereas bacteria which are deriving nutrients from a dead organism are decomposers, and bacteria which live in a mutualistic relation with a host is known as, with another organism is known as symbiotic, is showing symbiotic nutrition. Okay, come to the classification of bacterium. Next is how are bacteria classified? You know the sixth kingdom classification. Classify bacteria under Archaebacteria and Eubacteria. Eu means true. These are the true bacteria. Then what are Archaebacteria? So when we classify bacteria, we can classify them into Archaebacteria and Eubacteria. Archaebacteria, archae means ancient. Archaebacteria, ancient bacteria. Okay, that means they are also present now but under extreme habitats. These are the bacteria which are not in the common habitat. They are surviving in extreme climates. How can they survive in extreme climate is because of the nature of they contain specially hard cell wall deposition. So they can survive in the extreme climates and they are known as Archaebacteria. The bacteria which can survive in the extreme climates because of the specialized cell wall is known as Archaebacteria. Which are they? Mainly there are three groups. The first one are halophils. Halophils. The second one, thermoacidophils. Halophiles, thermoacidophiles, and the third group, again more groups are there as per your NCERT book. The next group is methanogens. Okay, the three types are halophils, thermoacidophils and methanogens. Then just have the idea, where are halophiles seen? Halophils are the bacteria inhabiting saline habitats. Okay, see, they are inhabiting saline habitat, whereas thermoacidophils are the bacteria seen in extreme hot, high temperatures. Okay, and highly acidic, high temperature regions. Then methanogens are the bacteria seen in marshy areas, marshy lands. Okay, and methanogens are also seen in the gut of other organisms, gut of cattle. You know, methanogens are present in the gut of cow like cattle. Okay, and this methanogens, okay, when this cattle excreted cow dung or cattle dung, when it is excreted, this cow dung consists of methanogens, and this methanogens bacteria release methane. Okay, this can release methane as a part of metabolism. Methanogens can release methane. So this cow dung is used as or the cattle dung is used as a source of biogas. Okay, we will discuss that in detail. Okay, anyway, not now, it's coming in another chapter next year. So methanogens are releasing methane gas as a part of their metabolism, so they can be used in the production of biogas. Okay, methanobacterium is an example. So methanogens are the bacteria which are seen in marshy lands and also seen in the gut of cattle. Okay, simple fact. That's about the classification of bacterium. This is giving a very brief classification of bacterium. A lot variety of bacteria are there. Different sessions are also there. But your book is giving a brief account, that is enough. Eubacteria we will discuss later. Archaebacteria, ancient bacteria, they belong to extreme climates and they can survive in the extreme climates due to the specialized cell wall. Then highly thickened cell wall. They are classified to halophils, thermoacidophils and methanogens. Okay, halophils in the saline habitat, this is an extreme hot climate, and methanogens in the two habitats you have studied. Okay, I think this is enough for today, and the remaining session Eubacteria we will discuss in the next class. Okay, thank you for listening. Thank you.